Hi dear students, myself Dr. Mashur, Head of the Department of Biotechnology, EMA College of Arts and Science, Kondoti. Welcome to my class. Today I would like to deal tools in biotechnology. We already told that biotechnology means application of technology into the field of biology. That means the application of scientific and engineering principles into the biology to make or modify a product, or to improve plants or animals, or to develop microorganisms for specific uses. This is the biotechnology. And in the last class we discussed about the different areas of biotechnology. There is animal biotechnology is there, environmental biotechnology is there, medical biotechnology is there, food biotechnology is there, and marine biotechnology is there. Like that, so many areas is there in for the biotechnology. For that, for the application in biotechnology, we have to use some tools in these different areas of biotechnology. In this class, I would like to explain the different tools used in this different areas of biotechnology. The first tool is the selection. Selection is the common tool used in biotechnology or some other field also using this selection. Selection is as a tool. Artificial selection is used to select the desired breeds of animals and plants to cultivate them for a better quality and quantity. And selecting microorganisms for desired products like bacteria or fungi producing specific enzymes, and antibiotic for selecting antibiotic producing microorganisms, and also selecting the biopolymer producing microorganism. And the selection procedure is also used for to select the microbes with improved quality or quantity. Like we can select the salt tolerant microbes or salt tolerant plants we can select, and temperature tolerant plants or temperature tolerant bacteria we can select, and we can select the microbes or plants with a high volume that will produce high volume of the end products or other biomolecules. This is about the selection tools which is used in the biotechnology. And the next tool used in biotechnology, that is a mutation. A mutation, we know that, is the sudden heritable change occur in the genetic material, and the process making mutation is known as mutagenesis. And this mutagenesis can be used to improve the quantity or quality of the products that will be produced by organisms. And the microbes with the enhanced production of biomolecules is synthesized by adapting random mutagenesis. This random mutagenesis can be used for the increased antibiotic production. And site-directed mutagenesis, there is a mutagenesis done at a specific site, and this SDM or site-directed mutagenesis can be used to introduce specific changes in a particular genes. And this site-directed mutagenesis can be used for enzyme engineering, or like that protein engineering we can use this SDM. And repeated rounds of mutagenesis and selection have been used to produce novel strains of microbes like bacteria, fungi with the improved characteristics. For example, novel strains that is producing improved amount of antibiotics and high amount of enzymes, and novel strains of microbes with improved or increased production of amino acids. This is the method of mutation. This is important in biotechnology. And the next tool used in the biotechnology is the fermentation. We know that is the process of conversion of sugars into the compounds such as lactic acid or ethanol by microbes. That means these microbes will use these sugars to produce compounds like lactic acid and alcohol. And in biotechnology, in industrial biotechnology, the industrial fermentation is the intentional use of fermentation by microorganisms such as bacteria, fungi, as well as the eukaryotic cells are also used for this fermentation, and insect cells to make product that will be used for human being. That means in industrial fermentation we are using microbes like bacteria, fungi, and human cells or insect cells to produce a specific protein or compound that will be useful for the human beings. This is the industrial fermentation. And using this industrial fermentation in biotechnology, we are producing acetic acids, citric acids, ethanol, enzymes, proteins, that will be therapeutic proteins, any other proteins, or we are using we are producing biofuels, biopolymers, etc. And these are the purpose of fermentation in the industrial. For the industrial fermentation, we can produce the biomass using the fermentation, and like a CPO, like the substances we can produce, and we can produce extracellular metabolites like alcohol or acids like we can produce. There's the extracellular metabolites we can produce. And third one, we can produce the intracellular compounds like enzymes, proteins, etc. And we can also use this fermentation technology for the transformation of substrate, in which the substrate molecule is changed to other products. That means there will be some change in the substrate molecule, and that will be transformed itself into the product. This is about fermentation. This is one of the important tool which is used in the industrial biotechnology. And the next tool is the tissue culture. The tissue culture is the main application in plant biotechnology, that also in animal biotechnology is important. Tissue culture is a method of biological research in that using fragment of tissue from animal or plant, and that will be transferred into the artificial medium in which they can continue to survive and they can do the function. That means we are taking a fragment of tissue from the plant or animal, and that will cultivate in the artificial medium for a specific function. This is a tissue culture. And the tissue culture may be consist of a single cell or population of cell or a whole part of a part of an organ that will be using in the tissue culture. And this tissue culture have an application in plant biotechnology for the production of high number of plants, and we can also use this tissue culture process for the production of transgenic plants, etc. And the animal cell culture have a wide application in industrial biotechnology and also in medical biotechnology. And in the industrial biotechnology, we can synthesize therapeutically important proteins using this cell culture. And we can also this animal cell culture has an application in tissue engineering, and we can use this for cloning, and we can use for the organ cloning using this embryonic stem cells or like stem cells. And we can use this cell culture for the production of metabolites, and like also we already told that we can use this cell culture for the production of therapeutic products. And this is one of the important tool that is used in biotechnology. And next tool is the genetic engineering or recombinant DNA technology, and this is the one of the tool that is used in almost all areas of biotechnology. Recombinant DNA technology means In this case, we are recombining the DNA molecules. That means the recombinant DNA technology or genetic engineering is the deliberate and the controlled manipulation of the gene in an organism with intent to making of that organism better in some way. That means if this genetic engineering will modify the genes present in an organism that will express or in a better way that will be used for the human being. That is the recombinant DNA technology. And the recombinant DNA technology is the branch of biology which is dealing with the splicing and recombining of the specific genetic units from the DNA of living organism as in order to produce new species or biochemicals, and that will be used for the human beings. Like we are producing insulin in bacteria, and we are synthesizing or producing transgenic plants and animals. And in genetic engineering, there are different steps. Is the first we have to take a vector, and we have to cut the vector using a restriction enzyme, and this restriction enzyme is known as molecular scissors, and we can insert the foreign desired DNA fragment into the vector, and that will form a recombinant DNA molecules. And for the after inserting in the DNA molecules in the vector, we have to ligate that DNA molecules in this vector molecules by an enzyme called ligase. And this recombinant DNA molecules will transfer into bacterium or host cell. For that, we are using so many gene transfer techniques like transformation, microinjection, biolistics or microvegetal bombardment, and electroporation like methods we are using to transfer genetic material into the host cell. And this recombinant DNA will enter the host cell, and from there it will multiply, and it will produce a number of copies of this DNA molecules, and this organism will be transgenic. That will be the host cell, that will be organism will be transgenic, that will produce new proteins. This is about genetic engineering. In genetic engineering, we are using some enzymes like nucleases to cut the DNA in DNA, and another enzyme, restriction enzyme, that is a restriction endonuclease, that will be specific to some site. Restriction enzymes only cut the specific site of the DNA. And the other one is the polymerase, and the polymerase, the DNA polymerase. DNA polymerase or RNA polymerase also is the DNA polymerase is the enzyme that is used to make the multiple copy of a specific DNA, and that they are using Taq DNA polymerase in PCR. And this enzyme is isolated from the temperature resistant bacteria named Thermus aquaticus. That's why this name Taq DNA polymerase. Yeah. And the next one is the DNA ligase. DNA ligase is also known as molecular glue, and this will use to ligate the DNA or join the DNA. And the alkaline phosphate is the enzyme that is used to remove the phosphate group from the end of the DNA. And in RNA technology, we have to use the vectors. Vectors are the molecular vehicles that will use to carry foreign DNA molecules to host cell. And the next one is the host cell, that is the cell that we are transferring DNA into that cell, that is known as host cell. And for this, we already told that we are using gene transfer techniques to transfer foreign DNA in RNA technology. There is some the gene transfer techniques that is used in molecular RNA technology. There is a transformation. In this, we are using heat shock method of transformation. And next one is the electroporation. Here we are giving electric shock to the cell, host cell, and due to the electroshock in a microsecond, that will create pores in the membrane of the organism. In that pore, this foreign DNA will be entered. And next one is the micro injection. In this gene transfer technique, next one is the micro injection. In micro injection, this small using the small needles or micro needles, we are injecting the foreign DNA the host cell. And next one is the particle gun. Here we are the machine will shoot the foreign DNA molecules that is combined with a microcarriers that will shoot the foreign DNA molecules into the host cell. This is about the genetic engineering or recombinant technology tools which is used in biotechnology, and this is the important one of the important tools that is used in biotechnology, because of almost all field of biotechnology that is using this recombinant DNA technology. The next tool is the nucleic acid hybridization. Nucleic acid hybridization is a phenomenon in which the single stranded DNA or RNA molecule will be annealed to its complementary DNA or RNA. That means when the double stranded DNA is denatured and it form a two single stranded DNA, and this single stranded DNA can able to bind and form hybridize and form a double stranded DNA, and this is the nucleic acid hybridization. And this phenomenon, this principle is used in to detect presence of specific sequence in a genome of an organism. For example, we know that genetic disorders or genetic disease caused by due to the alteration in the genetic structure of an organism or the DNA sequence of an organism. To detect the presence of this defective DNA sequence in an organism, we can use this nucleic acid hybridization. For that, we will denature the organism DNA and we will add a complementary DNA sequence that will be complementary to the defective part of the DNA sequence, and that small DNA will be labeled with the fluorescent dye or a radioisotopes, and this small DNA fragment will be bind with the specific DNA fragment specific DNA sequence present in the target DNA. If it will bind with the DNA, that will produce fluorescence or radiation, so we can understand that this defective gene sequence is present there. And this nucleic acid hybridization technique is used in southern blot for this using to detect the specific the DNA sequence in a DNA, and northern blot to detect the RNA, and fish. This is a fluorescent in situ hybridization. Is this fish is doing indirectly into the cell. This is about the nucleic acid hybridization. And the next tool in biotechnology, that is nucleotide sequencing. And this tool is used to detect the nucleotide sequences present in a DNA. And in the first generation, there is a two method were developed for the DNA sequencing. That is, one is the chemical method or Mason-Gilbert technique. This is a chemical method used for the DNA sequencing. And the second one is in the first generation, that is an enzymatic method, and is also called as chain termination method or dideoxy method. This is the second method in the first generation. And after that, first generation, there is one next generation sequencing is came, and this next generation sequencing enable many DNA fragments to be sequenced at one time and are most cost efficient and much faster. And these are the technique used for the nucleic sequencing. In the first generation, there is a two methods, a chemical, or there is also Mason-Gilbert method, and in that case chemical, we are using chemical degradation method, and the second one is the enzymatic method or chain termination method or deoxy method. And after that, after first generation sequence, there is one method, is the next generation sequencing, and this next generation sequence enable many DNA fragments to be sequenced at one time, and this is the more cost efficient and much faster. And this DNA sequencing can be used to find specific genes present in a DNA, and is also used for the identification of organism, or for DNA barcoding we can use this nucleotide sequencing. And we can also use this technique for these tools to discover genes involved in diseases. We know that some genetic diseases formed due to the presence of some abnormal nucleotides or unusual nucleotides present in some genes. That's why some genetic diseases formed. So that type of gene, the disease causing gene, we can isolate using this nucleotide sequencing. And using this nucleotide sequencing tool, we can get a better understand of a genomic structure and diversity among species, because this DNA sequencing we can use to create the phylogeny of an organisms. And this is the DNA sequencing tool. This is one of the important tools used in the biotechnology. And the tool is used in biotechnology, that is a PCR or polymerase chain reaction. There are different type of PCR is there, and the PCR technique is first developed by Carey Mullis in 1980s. That was one of the breakthrough research in the case of biotechnology and the molecular biology. And this technique is used to amplify the specific part of DNA, and that's why it is important in the case of gene cloning. And for the PCR method, we have required some components, like we need a DNA template that have to be amplified, and we need DNA polymerase that is used for the DNA replication, DNA amplification. In normal cell, this DNA polymerase is used by cell by its own DNA polymerase to replicate this DNA. And here in the PCR we will use tag DNA polymerase for the amplification, and this tag DNA polymerase is isolated from Thermus aquaticus bacterium, that is a thermal resistant bacterium. And we have to use nucleotides. Nucleotides are the building block of the DNA or nucleic acids. And we have to use a primer. There is a short piece of single stranded DNA that is complemented to the target sequence, and this primers will be complemented to end of the target DNA. And that's the components required for the PCR. And there's a different steps is there. What the first one, we have to go for denaturation. First we have to separate the two DNA strands, and we'll then next step is the annealing. In that annealing, these primers will be bind in the this complementary part, and then after that this DNA polymerase, tag DNA polymerase, will be undergo for the synthesis of DNA strand, that is known as extension. And this polymerase chain reaction is used to detection of and the diagnosis of infectious disease, and is also used in RDNA technology and used in DNA fingerprinting. And presently we know that if for the COVID-19 detection we are using this real-time PCR. That is one of the one type of an PCR. And this is the polymerase chain reaction tool. These are the major tools used in biotechnology, and many other tools also used in biotechnology, but these are major tools used in biotechnology. And I hope you understood this class. Thank you for the listening.